then it was just a lot. Like, there was just a lot of stuff. So
Yeah,
I know I had a couple of questions.
the kidney is, there's lots of parts
Yeah.
and a lot of details about how the parts work. You don't really understand
Yeah.
the whole picture until you put the
Mhm.
parts together.
Yeah.
And as you're studying the parts, you have no clue how the whole thing works until you learn the
Yeah.
parts. That's the thing that drives students nuts with the
Yeah.
kidney.
So it was all good, and then I'm trying to get.
Well, okay, well then let's kind of start from the very beginning. A very good place to start. Sounds like a song. Um, you had A&P, your HES, your anatomy is
Yeah.
probably very good. You don't need to
Yeah,
know
that part was all
where
fine.
the kidney is or what it looks like or
Yeah.
what have you. That's
Yeah.
probably fine. So
Mhm.
the nephron. Do you have any
Mhm.
questions about the structure of a nephron?
I think I did. I'm trying to find it. Mm, mm, I can't find it.
Is that your hair color or do you highlight your hair?
Yeah, the lighter part. This part is what my hair color is.
Okay.
The dark.
My wife wants to steal your hair
What?
because she's thin. Her hair is thin. It's fine.
Mm-hmm.
She always wished she had really thick hair. So
Yeah.
she saw your hair. She was in the gym once. She
Oh,
said, I want her hair.
oh. I would. Mine used to be a lot thicker than it is now, but I wish it.
College. You're pulling it up.
Yeah. Well,
My class, you pull, yeah. Okay.
no, but I dyed it, and now it's just thinner. I can't find the part in my PowerPoint because I. Oh, here it is. So there's the two types, the cortical and the juxtamedullary
Yeah.
nephrons. So do they do different things? And one has the shorter loop of Henle and one has the longer.
Yeah. The cortical has a short loop of Henle. It only dips a little bit into the medulla. The
Mm-hmm.
juxtamedullary ones, the loop of Henle goes very deep
Yeah.
into the medulla. Those are
Mm-hmm.
the ones that give you the most filtration
Oh, okay.
as opposed to the cortical ones.
Okay. Okay. That makes sense.
That's the reason why in some women, as a complication of pregnancy, they get kidney ischemia and they have like a stroke in the kidney, but it's
Mm.
only in the cortex.
Hmm.
So you lose some kidney function, but you don't lose all your kidney function
Yeah.
because the medulla is spared. And so the juxtamedullary ones, which do most of the heavy lifting, they're still functional.
Yeah. Hmm. Okay. So is that the only difference between them? Just the
Yeah,
loop?
is the length of the loop of Henle
Okay.
and the location of the Bowman's capsule.
Yeah. Okay. Okay. Then next, you talked about filtration.
Think of a coffee filter.
Okay.
That's probably the best way to think of it.
Okay.
So, coffee filter, this is my mask, but it looks like a coffee
Yeah.
filter. So, you put the grounds in,
Mm.
you pour the hot water through, out comes essentially a distillate of the grounds,
Yeah.
and the grounds themselves can't get through the filter.
Uh-huh.
And so, you get a filtrate
Yeah.
of the coffee, of
Yeah.
the extract of the grounds. That's a lot like how the Bowman's capsule works. Yeah, the podocytes from the inside the Bowman's capsule sitting on top of those glomerular vessels. The glomerular vessels are full of pores.
My Wi-Fi. Sorry, you were breaking up for a
Oh,
second.
you keep also.
Yeah.
That better? You froze. Yep, you froze up there.
Okay. I'm back, I think. Okay, I think it's good.
I'm plugged in, so I'm doing this via ether cable, so
Yeah, I just switched it so I can hear you now.
good. Okay, would it help? Is our microphone strong enough, or should I use?
Yeah. No, I think you're fine.
Okay, all right.
It was just like freezing.
Okay. So the podocytes
Mm-hmm.
provide filtration, and I think this picture here does about as good a job as you'll ever get, showing essentially how that filtration works. So you have the
Yeah, I have that.
endothelial cells, then the basement membrane, and then the interlocking pedicels
Mm-hmm.
of the podocytes. They have small pores too, but this matrix is highly charged, so you can get ions, water, things soluble in water, proteins, too charged.
Mm-hmm.
Cells, way too big.
Mm-hmm.
So that shows you essentially how you get this filtration. And
Yeah.
this is driven by the pressure of the blood squeezing the fluid through there. Once you squeeze that fluid through there, now it becomes known as filtrate. And that then goes into the proximal convoluted tubule.
Okay.
So when blood pressure crashes, like during sepsis
Mm-hmm.
or something like that, or if somebody has a car accident, they have a really bad hemorrhage, they get a broken jugular, they get a compound fracture, the blood pressure because of blood loss is going to weigh down, and your kidneys will stop filtering because they don't have that pressure
Yeah.
driving the filtration. That filtration, by the way, you can measure. It's called glomerular filtration rate, or GFR.
Yep.
GFR.
Yep. I remember that. Um. And that's measured per minute, right?
Yeah. Per unit volume per minute. So
Yeah.
milliliter per minute.
Um, and then
Proximal convoluted tubule. You know how that works?
I remember talking about it. You talking about it.
Can you explain to me how it works?
That was at the end. It was that in the second lecture?
No, first one.
Because there's proximal and distal. I remember talking about that.
Yeah. Proximal and distal with the loop of Henle in between.
Yeah. I don't know where it is. Well, no, I don't think I can explain it to you.
So when the filtrate goes through the PCT and it gets to the end of it, it has the same osmolality as when it started. What
Yeah.
does that tell you?
Mm, I don't know.
Tells you that the amount of salt you're reabsorbing and the amount of water you're reabsorbing are equivalent.
Okay. Yeah.
So you've decreased volume, you haven't affected the concentration.
Yeah. Okay. That makes sense. Oh, here.
There we go. So what's the structure of the epithelium of the PCT?
The pump.
So it's a cuboidal epithelium,
Mhm.
but the apical surface is full of microvilli.
Okay.
That's why I've drawn these
Hmm.
really ugly looking ridges here. And then what's on this side? This is the basolateral side.
Hmm.
Remember, it's a epithelium. So all epithelia sit on a, what's it called?
I don't know.
Yes, you do.
Hmm.
Yes, you do. Basement, basement. So I kind of drew that little
Hmm.
basal membrane. Basal membrane. Okay. So this is the basolateral side.
Hmm.
This is the apical side. The filtrate is here.
Hmm.
And it's as though we've taken the tube and kind of cut it cross section like this.
Hmm.
Okay, then what else is, what else, what else is over here?
I don't know. I'm not.
Are you a visual learner?
I don't know.
Because drawing pictures, if you're a visual learner, drawing pictures of things will help you learn it.
I think I need to when I physically do things, it helps me learn it.
Okay, which will help you in PT school
Yeah.
because you'll do stuff and
Yeah.
it'll be muscle memory
Uh-huh.
and you'll know how to do it. Then and I would suggest just go ahead and sit there and doodle
Draw
and
them.
draw yourself pictures. By drawing pictures, it will cement it in your mind
Okay.
because you're an athlete, so you're a muscle memory kind of person.
Yeah.
By drawing stuff, you'll learn it, you'll remember it better. And it
Okay.
doesn't have to be pretty. Good night. Look at my pictures. So I'm going to put over here, I'm going to use a different color. Okay. See that in pink?
Hmm.
That's a blood vessel, right?
Okay.
Okay.
Hmm.
We'll go ahead and label it. Okay. That better?
Mhm.
Okay, so what happens to the filtrate in the PCT? What do the PCT cells do to the filtrate?
It takes stuff out of it and puts stuff in it.
Um, that's basically right. Can you give me a few more details?
Um,
It's kind of like my Bible quizzers. They'll say, Jesus. Well, yeah, that's correct, but can you give me more details?
there's a lot of details.
Well, and the details are important.
Yeah.
Details can change the diagnosis, can't they?
Yes.
So you have to pay attention to them. So let's pay attention to details. All right, let's start with glucose.
Okay.
Glucose. You want to recapture glucose?
What?
Do you want to recapture glucose, or do you want glucose to just go out the drink?
You want to recapture it?
You want to recapture it. That's an energy source. You bet your life you want to recapture it. So we're going to use a different color. We're going to take sodium, and we're going to bring in sodium and glucose. So we're going to use secondary active transport. This cell has a low sodium concentration inside, right?
Mm-hmm.
Why?
Because there's none in there.
That again, that's correct, but why?
I don't know.
Where's that picture? They took that picture out of this particular version. Okay, so on the I'm running out of colors. Okay, on the basolateral side, there's a ion pump. All cells have this ion pump.
Mm.
It's the sodium, potassium, ATPase. What does it do?
Takes out the sodium
Yeah, and puts it where?
outside.
To what here?
Yeah, there's no
Yeah.
sodium in it.
Okay, so you can kind of sort of draw a straight line from the filtrate to the bloodstream, can't you?
Yeah.
That's pretty much what happens. So again, if you draw pictures, sometimes drawing pictures will clarify things and help you learn it.
Mm-hmm.
All right, so oh boy, that in focus, or can you see that at all? Or
Yeah.
all right, so glucose comes in via sodium. There's a secondary active transport. Because sodium here is low in this cell, sodium can come in down its concentration gradient. We can use that energy to bring glucose into the cell with it. Sodium will then be removed from the cell via primary active transport. The ion pump on the basolateral surface will take the sodium and put it right back into the blood at the expense of potassium. And this is a primary active transporter, so it'll hydrolyze ATP.
Okay.
What about the glucose? So glucose there is in the basolateral membrane a facilitated diffusion glucose transporter called T1 or sometimes T2. And as glucose increases here, it now goes from the tubal cell straight to the blood. So glucose then goes straight from the filtrate to the tubal cell back into the blood. So what have you done? You've recaptured glucose, you've recaptured sodium, and water comes along for the ride. See how that works? So it's a combination of secondary active transport at the apical side, primary active transport, and facilitated diffusion at the basolateral side, all to bring these materials back into the bloodstream. And it does that with glucose. It also does that with amino acids. Also does that with calcium. Does that also with phosphorus. And it will also do that with magnesium. So you recapture using this type of strategy all these things you want to keep: amino acids, glucose, and then electrolytes. Water goes with it, and you reabsorb 65% of the water and solutes in the PCT, regardless of your hydration status. All right, let me pause right here. See if there's any questions.
No, that was helpful.
That was helpful?
Yeah.
So if I asked you to explain it to me, you could?
I think.
You think?
Yeah.
Okay, do me a favor. Do you have a piece of scratch paper nearby?
Yeah, I can grab one.
Can you draw it out for me? And talk me through it while you draw it out? Go ahead, do that right now.
No, I can do it on here, actually.
I think you'll find it's helpful if you draw it out.
Yeah. Well, because you drew them on the lecture videos, and I was trying to keep up, but I was struggling.
You know you can pause them, right?
I know, I did. Okay.
Students sometimes think there's shame if they pause the video. There's no shame in pausing it.
I know.
Okay, talk me through it. Go ahead.
Okay, I'm going to draw. I don't know how to show you.
You don't have to show it to me. Just draw it, show yourself, and you can show me when you're done. Go ahead.
So I'm going to draw the cells, the cuboidal cells. And this side has microvilli, so I'm going to do ridges. And ridges. And the cell. Separate them. And the filtrate goes down through the middle. It's got all the stuff in there. And
stuff? What stuff? Be specific. What stuff?
Glucose, sodium, calcium, phosphorus, amino acids. These are all things that will be reabsorbed through the cell.
Yep.
First through secondary transport, active transport.
Yep.
What is this side called with the villi?
The apical side.
The apical side.
The apical side. The side that faces the filtrate is the apical side.
Okay.
If you're a cell, apical is up.
Oh,
Look
okay.
at pat the top of your head. Okay, that's your apical side if you're a cell. Look down at your feet.
Mhm.
That's your basolateral side if you're a cell.
Okay.
Okay,
Mm-hmm.
that help?
Mm-hmm. So when the stuff is reabsorbed, it'll go in through the apical side through secondary active transport into the cell. And then, oh, I forgot to draw the membrane on the basal side. Membranes, and then outside of those are the blood vessels.
See, you're getting it. You're getting it.
Mm-hmm.
Keep going.
Okay. So you get, say, glucose through the secondary transport into the cell, and then to get out into the blood vessel, it goes through primary transport.
No, the glucose is transferred via facilitated diffusion.
Oh, so that's T1 or T2.
Yeah, that's the GLUT1. GLUT, I said T1, I'm sorry. GLUT1 or GLUT2.
Okay.
GLUT1
Yeah.
or GLUT2.
Okay.
They're both glucose facilitated diffusion transporters.
Okay.
And they're not induced by insulin. GLUT4 is the one that's induced by insulin.
Oh, yeah, I remember that. So that's just for glucose.
Yeah.
Okay. Glucose goes through there. And so then for, say, sodium, goes through first through the secondary transport with water
Yep.
because the cell has low sodium. And then it'll go through the primary transport, oops, out into the blood vessel.
Okay, when you're done, you can show it to me. And oh, that's an iPad.
Uh-huh.
Oh, that's cool.
Something popped up.
Good job. Good
Yeah.
job. Okay. Okay. Did that help?
Yes.
I, in all honesty, Emily, I'd recommend doing this
Yeah.
each time. Draw it out
Mm-hmm.
because you'll remember it better
Yeah.
when you draw it out. Okay. Loop of Henley.
Mm-hmm.
Have you listened to that part yet?
Mm-hmm.
Do you understand how it works?
Mm,
It's not simple.
there's a lot of different,
Pieces.
yeah. Mm, the descending loop is permeable to water, impermeable to salt, and then the ascending is the opposite of that.
That's correct.
So, like, okay, the descending limb has a lot of the aquaporin channels, and that's why water goes out.
That's correct. But why does it leave? Why does it leave the tubule?
Mm, I don't know. Does it have to do with the like the osmolality of the
There you go.
medium?
What happens to the medulla? Because it's descending into the medulla.
Mhm.
What's the case with the medulla?
Like, why does it make it go down? The osmolality go down? Oh, the osmolality starts to increase
There you go.
as it
The
goes down.
water is moving down its concentration gradient.
Yeah.
So there's no energy required to move the water.
And then it starts to, once it gets to the bottom and go back up, it starts to go back down again.
What goes back down?
The osmolality. So then
Why?
when it gets back to the top, the concentration is the same.
Yeah, about the same as it was when it left.
Yeah.
Why does the concentration go down as it ascends the loop of Henle?
Because of the sodium chloride going on? Or no?
Yeah, good. Sodium and actually urea
Oh, yeah.
and some chloride too, for that matter.
Okay.
Why do they leave?
Because, well, so the concentration, as the volume decreases, the concentration doesn't increase. So it stays the same.
You confuse me. I don't know what stays the same.
The concentration of the filtrate as the volume of it decreases.
The volume decreases, but the concentration stays the same. That's the case for the PCT. That's not the case for the loop of Henle.
Oh, yeah. Okay. I don't
Take a
know.
look at take a look at figure 1714. Do you have your book on your computer?
I have, yeah.
Take a look at that picture.
Mm. I feel like I have to keep like referring back to the overall picture or else I get like lost
That's fine.
where I'm
That's
at.
fine. As the details become more familiar to you, you'll need to do that less and less. So don't
Yeah.
worry about that.
You said 17, 14?
Yeah, 1714.
13.
It should look
Okay.
like
Yep,
this.
I got it.
Okay. Look at that. Tell me what you see.
Um, the descending and ascending limb, the loop.
So what happens on the descending side? What do you see there?
H2O leaving, water leaving. As it goes down, the osmolality increases.
There you go, because you're depriving it of water only.
Okay.
So the osmolality goes up.
Okay.
So that
Yeah.
it's continually isoosmotic with the concentration of solutes in the medulla.
Okay.
Now, as it starts to ascend, what
Mm-hmm.
happens?
the sodium leaves, goes out, and as it goes up, the osmolality goes down.
There you go. That's correct. So it's now that stretch of the loop of Henle is impermeable to water. So water can't leave.
Hmm.
But what does leave? Salt.
Yeah.
Salt. That's why the filtrate becomes less and less concentrated because now salt is leaving.
Yeah.
Does that make sense?
Hmm.
And then when you get to the thick ascending loop of Henle, now it actively transports salt
Hmm.
from the filtrate.
Hmm.
And that's actually what's going to maintain the gradient of salt in the medulla that will actually help this whole thing happen.
Yeah.
Also, the other piece there in that picture, the water that leaves doesn't dilute the concentration of solutes in the medulla because the water is taken up right away by the vessels, by the peritubular
Yeah.
vessels.
Okay. Got it.
Okay, now you repeat it back to me. You explain it to me.
Okay. So in the descending limb, as the filtrate goes down, the osmolality increases and water leaves and is absorbed by the vessels right away. And
Good. Keep going.
so it keeps going down, and then as it turns to go back up into the thin ascending and then the thick, salt will leave once it gets to the thick part of the ascending limb, and the osmolality will decrease, will go down as it goes up.
Very good. Very good. You have accurately recapitulated how it works. By the way, that pump that's in the thick ascending loop of Henle,
Mm-hmm.
the NKCC2,
Mm-hmm.
we're going to talk about when you take pharm,
Mm-hmm.
either next year or next semester, whenever you take it.
Yeah, in the spring.
We're going to talk about a group of drugs called loop diuretics. That's the target for loop diuretics, is the NKCC2. So no, you don't escape this so quickly. It'll come back to haunt you.
Okay.
Aren't you happy? Okay. Then we get to the DCT, and the DCT has two parts, early and late.
And that's the distal.
Convoluted tubule.
I remember talking about that. Do we want to talk about the vasa recta that surrounds the loop of Henle?
The loop of Henle, the peritubular vessels that surround the loop of Henle are extremely branched and very delicate, and they're called collectively the vasa recta.
Okay.
They're the ones that reabsorb all that water.
Okay, that makes sense. Okay, that's not. Let me find.
Okay, so the early DCT is impermeable to water, just like the thick ascending loop of Henle.
Mm-hmm. Okay.
So it contains more sodium than the tubule cells. So sodium flows down its concentration gradient into the tubule cells because of various protein channels for sodium. And these channels are cotransporters. So they cotransport sodium and chloride. They're sodium chloride cotransporters. And remember, in the DCT, we have a nice cuboidal epithelium, no microvilli.
Okay.
That's an important difference between the two.
Yeah.
And I think when I did the drawings of them, I left out the microvilli.
And
I
the
hope
second
I
one?
did.
I'm not
I may have put them in by mistake. If I did, shame on me. There's no microvilli in the DCT.
okay.
So you have the sodium chloride cotransporters. So you reabsorb sodium, you'll reabsorb chloride. Sodium goes right back to the bloodstream via the sodium potassium ATPase. And then chloride will leave, because chloride is very low in the blood, it leaves straight through a chloride channel and back into the bloodstream. Then there are sodium calcium channels. Sodium calcium channels. So they move calcium into the tubule cells with the exchange of a sodium. With the exchange of a sodium. So the sodium moves from the tubule cell into the filtrate. Calcium moves from the filtrate into the tubule cell, and then it moves across the basolateral membrane into the bloodstream.
Mhm.
All right?
So
And
because,
go
sorry,
ahead.
are there those like different channels because there isn't that villi? Like, is that why things are transported through the cells without the
No,
villi?
the DCT is much more specialized
Yeah,
in what it recaptures. The PCT recaptures
just
tons of things.
okay.
Amino acids, glucose,
Mhm.
phosphorus, lactate, um, bicarbonate, lots of things.
Okay,
DCT, much more specialized.
okay.
And the DCT is subject to control by hormones.
Oh,
So remember when we talked about the endocrine system, we talked about parathyroid hormone?
hmm,
That controls calcium reabsorption in the DCT.
okay.
So the parathyroid hormone increases the sodium calcium channels in the DCT that increases calcium reabsorption, thus raising calcium levels in the blood.
Hmm.
When do you make parathyroid hormone? When calcium levels drop.
Okay, that makes sense.
So again, draw a picture, draw it out. In fact, do it right now. Do it right now. Go ahead and draw DCT cells, leave out the microvilli.
Okay. Filtrate. And it still has that membrane on the.
Basement membrane, yeah. Emily, all epithelial layers have a basement membrane.
Okay.
That's the very definition, well, one of the definitions of an epithelium. Epithelia cover things, they
Yeah.
line things,
Hmm.
and they always sit on an epithelium, on a basement membrane. Some are thick, some are thin, some contain collagen, some contain fibronectin, some contain fibronectin, some contain laminin.
Just
Depends.
depending on where they are.
Right. It's kind of like, think of it as your tennis shoes. Some have Nike, some have Adidas, some have Saucony's, some have J.C. Penney's. You know, we all have tennis shoes.
Hmm. Okay.
So on the apical side, you have a sodium chloride cotransporter.
Hmm.
And then on the basolateral membrane, you have a sodium potassium ATPase.
Okay, so what is it? Like, what's something that will be filtered through in the DCT?
So sodium and chloride is recaptured,
Mm-hmm.
and calcium is recaptured.
Okay. And they're all, like, each different one has its own channel.
Yeah, yeah.
Okay.
Ion channels have been extensively studied. They're specific.
Okay.
Big exception is the nicotinic, the ligand-gated nicotinic acetylcholine channel. It lets in cations pretty much.
Okay.
It's not very specific. Most other channels are very specific.
Okay.
And then you have a calcium channel on the apical surface
Mm-hmm.
and a calcium sodium exchanger on the basolateral surface.
Okay.
And that calcium sodium exchanger, go ahead and underline that one and just remind yourself the synthesis of that one is upregulated by parathyroid hormone. And I don't know why, but your camera goes light, dark, light, dark, light, dark.
I think it's because I'm moving
Oh, that's why.
and
Okay.
it focuses on the window.
Oh, that's probably why. Okay.
Parathyroid. Okay, so calcium
Okay, show it to me.
is in the filtrate, goes in.
I can't see it.
Oh, you want me to show you?
Yeah, show it to me. Yes. Show me your gorgeous artwork. Okay.
So
Sodium and
calcium.
chloride.
Uh-huh.
Okay. Draw some arrows. Show me how sodium and chloride go in.
Should have made them different colors.
All right, let's see. Yes. There you go. And there, okay. Just make sure you put in the bloodstream there, the ions going into the bloodstream so that you know they don't go out into outer space, they go back into the blood. So reabsorption brings things back into the blood.
Okay.
That help? There you go. Good job. Good job. Okay. Now we go to the late distal convoluted tubule, which is a lot like the collecting duct. And there are two specialized cells. What are they called?
Mm-hmm.
He was your buddy in high school? Or maybe she. If you got in trouble, you got sent to her.
Principal.
Principal. Thank you. You're probably going to say vice principal.
Yeah.
Principal cell. What's the other one?
I don't know.
Alpha. It's put in between the epithelial cells, so it inserts between them. It intercalates in between them, so it's the alpha
Inter.
intercalated cell.
Okay.
Alpha intercalated cell. INTERCALATED cell. Intercalated cell.
Okay.
Want me to spell that again?
No, it's okay.
Got it? Okay, good.
Okay.
All right, so this has the principal cell has two channels on the apical surface. It has the ENaC. What's the ENaC?
Has
It's a
on the
sodium channel. ENaC stands for epithelial sodium channel.
okay.
That's why it's ENaC, ENaC.
Okay.
And then a potassium channel. So sodium flows into the cell down its gradient. In order to electrically balance what's going on in the cell, potassium flows out of the cell. The
Okay.
sodium goes in, potassium goes out.
Mhm.
Then on the basolateral side, we have our old buddy, the sodium potassium ATPase, that kicks the sodium back out into the bloodstream.
Okay. So when you said sodium goes in, potassium goes out.
Yep,
When you say it goes out, does it go out?
into the filtrate.
Okay. And does this it looks the same as the early
Right, no microvilli.
Okay. We'll
But
draw it.
functionally it behaves more like the collecting duct.
Okay.
And what does the alpha intercalated cell do?
Where did you say that was? Where is it at?
That's also in the late DCT and collecting duct. Oh, Emily, look out your window. Look at that sunset.
Oh yeah.
Oh, that is gorgeous.
Pink and purple.
That is just gorgeous. I just had to make note of that. It's
I
just
didn't
really,
even notice.
it's very pretty.
Okay.
What does that cell do? The alpha intercalated cell.
Mm, I don't know.
Hydrogen. Gets rid of hydrogen ions by pumping them across the apical surface into the lumen of the nephron.
Okay. So it takes them from the filtrate?
No, from your blood.
Oh.
So it has a hydrogen ATPase. So there's a primary active transporter, pumps hydrogen from the cell into the filtrate.
Uh-huh.
Excuse me. Then it has a hydrogen potassium ATPase that pushes out a hydrogen in exchange for a potassium, both against their concentration gradients. And the late DCT and the collecting tubule are targets for the hormone aldosterone. What makes aldosterone?
What makes it?
Yeah. Does your big toe make it?
No.
Do your eyeballs make it?
Mm-mm.
I'm being facetious, obviously. What makes it?
The adrenal cortex.
Very good. Which layer of the adrenal cortex?
I don't know.
Outermost layer. Zona glomerulosa. Say it.
Zona glomerulosa.
That's the layer that makes aldosterone.
Okay.
Okay. What is the target for aldosterone? The target are the principal cells and the alpha intercalated cells.
Okay.
So aldosterone increases expression of the ENaC
Okay.
and the basolateral sodium potassium ATPase.
Okay.
In the alpha intercalated cells, it increases synthesis of the hydrogen potassium ATPase and therefore increases hydrogen ion secretion.
Okay.
Draw that out. Go ahead, draw it out on your iPad if that helps you remember it. Go ahead and draw it out. Draw aldosterone. Remember, aldosterone's a steroid, so it charges right through membranes. Doesn't have to bind a receptor on the surface. Comes right through the membrane.
So draw it going from
The bloodstream, because it's an endocrine
through,
hormone.
just straight through the cell.
Yep.
Okay.
And so you get increased ENaC, increased sodium potassium ATPase. What does that mean? You're going to absorb more sodium.
Okay.
You'll absorb more sodium at the expense of potassium. And if you have hyperkalemia, your adrenals make more aldosterone to help you get rid of more potassium.
Okay.
Right? Okay, one more piece. Principal cells respond to anti-diuretic hormone.
Okay.
Did you get that piece? I tried to draw it out where the aquaporins are in little vesicles underneath the apical membrane.
I think so.
So those vesicles are full of aquaporins. When ADH binds a receptor on the basolateral surface, those vesicles fuse with the apical membrane, and now there's more aquaporin, so you take up more water from the filtrate. You
Okay.
recapture more water, you make a more concentrated filtrate, and you produce less urine. Helps you conserve water.
Okay.
You look like you're ready to cry.
Mm-mm.
No?
I'm just trying to put things
That's
together.
your thinking pose. Okay.
I'm trying to put things together. I'm trying to see it big picture, I think. Bigger.
Well, when you look at a kidney,
Uh-huh.
and hopefully you never look at a kidney the same way anymore,
Probably not.
think of it as a huge collection of nephrons,
Okay. Yeah.
because a nephron is the functional unit of the kidney.
Uh-huh.
And then think of the nephrons in terms of their parts and how each part works to recapture things from the filtrate that you want to keep, so that you produce a highly processed filtrate that gets rid of what you want to get rid of, but keeps what you want to keep.
Okay.
and then varies that according to your hydration levels.
Mm-hmm. It's crazy.
It's elegant. I hope you can see there's
Yeah.
also an elegance to it.
Yeah.
All right. Did
Yeah.
Kayla give you any questions to ask?
No.
I think
She
maybe she was hoping that whatever we talked about would kind of hit her questions.
probably. I mean, I think just going over and like each section and redrawing it was very helpful for me.
I'd encourage you to do that,
Yeah.
to draw things out,
Yeah.
because I think you're a visual learner.
Yeah.
At least that's what Beth Ulrich tells me. She says you're a visual learner. There's nothing
Yeah.
wrong with that. Most people are. So
And I think
drawing
it's better
pictures,
when I do it even more than seeing someone else draw it.
well, then do that.
Yeah. I
Do
will.
that because it helps you learn it. And when I draw it out, it helps me teach it.
Yeah. I bet.
Okay.
Mm-hmm. We have a quiz on chapter 17 tomorrow.
It'll
Okay.
be online. It'll be on
And
Blackboard.
then, yeah. And
That'll
then
be your
I
last
heard,
quiz.
yeah. Okay. You said in the lecture you were going to make a practice test probably over the weekend.
Yeah, if I have a Bible quiz Saturday, so my Saturday's pretty much shot.
Okay.
So I'll take my computer home. My wife hates it when I work on Sunday, but have to do it.
Mm.
She's Dutch, so she came from the Dutch Reform tradition. They have a really strong Sabbath tradition in that church, which is good.
Yeah.
It's a
Yeah.
good thing to have.
Uh-huh.
So she won't do laundry on Sunday. The girls needed her to shop once on Sunday, and she
Mhm.
did it very begrudgingly because she said, Sunday, you don't shop on Sunday. You
Mhm.
don't do laundry on Sunday.
Yeah.
Sunday's a Lord day. You do acts of service and you go to church and you study your Bible and you spend time with family and
Yeah.
blah, blah, blah,
That
and
is
all that.
good.
And actually, it's been good. It's been good for us
Yeah.
as a family because we get so busy during the week.
Yeah.
Sunday's been a good time to retool. So we've kind of kept that. So I'd have to tell her, hun, finals week. I
Yeah. This
have to
time
do
of
it.
the semester is rough.
So anyway, it's a Dutch thing, but that's okay. It's a good Dutch thing.
Yeah. Okay.
You're from that side of the state, aren't you?
No, I'm from Jackson.
You're from Jackson. Okay.
Mhm.
Some of our colleagues are from Grand Rapids, that area where there's a lot of Dutch people. And
Yeah, I think a lot of people, like students even, are from the west side of the state.
right, so they might have that
Yeah.
tradition because I was explaining that to somebody once and they, oh, yeah, 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 I grew up that way. Yeah, no, we didn't do that
Well,
on Sunday and what have you. So, all
yeah,
right,
Sunday was always like laundry and grocery day in my family. So
because you're catching up because
yeah,
the week was so crazy.
yeah, and it's like get ready for the next week. But
Yeah, it's kind of my mentality too. When I
yeah,
was growing up, because, when you're on a farm, you work 6 days a
yeah,
week.
yeah,
What do you do on Saturday? All the yard work you didn't get done during the week. So your
yeah.
Sunday, when I grew up, was yard work day.
Mm-hmm.
And Monday, you go back to work at
Uh-huh.
7 in the morning, 6 in the morning. If you're out in the fields, 5 in the morning. So
Wow.
life of the farmer, way it goes.
Yeah.
Digestion is fine. You have any questions about digestion?
No.
No? All right. So if you think of anything, just email me.
Okay.
I'm not hard to find.
Okay.
Okay?
Well, thank you. That was very helpful.
Good.
I
I'll
feel
get
better.
this recorded. Well, I'll get this processed. It'll go on my YouTube channel, and I'll send the link to Kayla.
Okay.
And if you see
Right.
her, I'll send you guys emails
I'll
with the
tell
link.
her.
Okay. All right,
Okay.
then. Have a good evening.
You too.
Study hard.
I will.
Make me proud.
Okay.
See ya.